Good morning, Mr. Taifo. Oh, good morning, Mr. Taifo. Good morning, Mr. Taifo. Oh,
Good morning.
good morning. Yeah, okay. Uh, I'm Silvina, one of the committee here. So please wait. Maybe in 5 minutes we will start our event today.
No problem. Thank you.
Thank you, Mr. Taifo.
Dreaming, I must be dreaming.
Mr. Taifu, yeah, we will start now.
I cannot hear.
Yang telah disediakan. Demi keamanan, dihimbau untuk tidak meninggalkan barang tanpa pengawasan. Hadirin tidak diperkenankan merokok di dalam gedung ini dan di kawasan kampus FMIPA UNS kecuali di area merokok. Pastikan puntung rokok mati pada saat membuang ke tempat sampah. Kita berada di gedung C yang memiliki 5 lantai dan saat ini kita berada di aula lantai 5. Apabila terjadi gempa bumi, lindungi diri Anda dengan meja atau kursi, lalu tunggu sampai keadaan benar-benar kondusif. Di setiap lantai gedung ini memiliki alat keselamatan berupa alat pemadam api ringan atau APAR yang terletak di setiap sudut ruangan. Jika terjadi kebakaran, hadirin diharapkan tetap tenang dan jangan panik, segera keluar melalui pintu darurat menuju assembly point atau tempat berkumpul yang aman. Penting untuk kita mengetahui prosedur evakuasi apabila terjadi keadaan darurat. Dimohon tidak menggunakan lift dan dengan tertib menuju pintu darurat pada gedung ini. Pintu darurat terletak di sebelah kiri panggung dan di belakang ruang aula, kemudian mengikuti jalur evakuasi dan berkumpul di assembly point dengan bimbingan petugas keamanan dan atau panitia. Assembly point terletak di lapangan basket yang berada di depan gedung C dan halaman depan gedung A. Dalam keadaan darurat atau jika ada orang atau benda mencurigakan, silahkan menghubungi 0271 669 376 atau petugas keamanan terdekat. Demikian induksi keselamatan kami sampaikan. Kami ucapkan terima kasih atas perhatian dan kerjasamanya. Utamakan kesehatan dan keselamatan kerja.
Ladies and gentlemen, as a mark of respect for our nation and institution, please rise for singing of Indonesia Raya, followed by March FMIPA Universitas 11 Maret. Please kindly stand.
Fakultas IPA Universitas Sebelas Maret junjung beliau guruan tinggi berbakti pada Ibu Pertiwi Mangestilu kau bangun negeri wawasan luas dengan keluhuran budi kembangkan ilmu dan teknologi melanda bersama FMIPA jaga jujur dalam mana-mana aktifasikan karya unggulan yakinkan jadi lembaga putra akselerasi menuju kelas dunia. Civitas Fakultas IPA Universitas Sebelas Maret junjung tinggi pada Ibu Pertiwi Mangestilu bangun negeri wawasan luas dengan keluhuran budi kembangkan ilmu dan teknologi sama FMIPA jaya jujur dalam mana-mana aktifasikan karya unggulan yakinkan jadi lembaga putra akselerasi menuju kelas dunia. Dance besar.
Thank you. Please be seated. Assalamualaikum warahmatullahi wabarakatuh. Good morning. Okay. Our distinguished guest, Professor Dr. Sigit Prastowo, Director of Academic Affairs of Universitas Sebelas Maret, representing the Vice Rector of Universitas Sebelas Maret, Professor Dr. Desi Suci Handayani, the Dean of Faculty of Mathematics and Natural Sciences, Universitas Sebelas Maret, respected speakers, honorable delegates, ladies and gentlemen. On behalf of the Faculty of Mathematics and Natural Sciences, Universitas Sebelas Maret, It is my great pleasure to welcome you to the International Shortcuts SDGs Equity Program 2026, entitled From Nature to Innovation, Exploring Sustainable Science in the Tropics. My name is Putri, and I will be your master of ceremony for today's event. Ladies and gentlemen, we are honored to invite the Vice of Rector Universitas Sebelas Maret, represented by Professor Dr. Sigit Prastowo, to deliver the opening remark and officially inaugurate the International Shortcuts SDGs Equity Program 2026. Professor Dr. Sigit, the floor is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. It is my honor to stand in front of you, and a very welcome to all distinguished speaker, participant, guests, then college and the university, the faculty management from Faculty of Mathematics and Natural Sciences. On behalf of university managements, I welcome you all to this nice opportunity, and as a great honor that you will involve in short course program 2026, with the title of From Nature to Innovations, Exploring Sustainable Sciences in Tropics. This short course will be held in 25 to 27th of June. Then within these 3 days, you will have a lot of sharing knowledge, and then we'll have a lot of new idea, which is might be can spark about future idea for specific research or specific collaborations. For sure, this short course held because of at this time we face a lot of critical moments that world facing climate change, environmental degradation, and many international problem, which is cannot be solved only with one person's or only with one subject, but we need to collaborate each other with many peoples, with many idea, with many abilities, and with a lot of things which is can support especially the SDGs goal, which is already underlined one of the universities plus Maret mark. The theme of this short course reflect that we all believe that science should be as the platform for solving problems. A lot of problem will be solved by science, because science will be as a great solutions, empowering communities, contribute to sustainable development goals. Especially when we are talking about the nature of biodiversity of Indonesia, we have a lot of natural products, we have a lot of natural compounds, and we have a lot of tropical resources that waiting outside there. We need to explore, and then later we can commercialize to help people and to improve the people life. In this nice opportunity, we also delight to welcome all the participants. I got the numbers from the faculty management, that's around 50 participants representing diversity of academic and professional background from different university and also from different country. I can mention here that There are participants from Malaysia, Pakistan, Afghanistan, Rwanda, Madagascar, and Nigeria. Then this representing that the importance of these international collaborations in this regard. Through science and through innovations, we can solve a lot of problems. And also, we are also equally honored to the expert who already joined in this short course. Maybe I can mention here in the online, I saw Prof. Taifo Mahmud from Oregon State University. Nice to meet you again, Professor Taifo. Then you are commonly come to Universitas plus Maret, especially when I'm still in Faculty of Agriculture. Then Prof. Muhammad Yusri bin Abdurrahman. This is from University Kebangsaan Malaysia. Dr. Nurdiana Samsurizal. This is from University Teknologi Mara, Malaysia. Then Mr. Rohaizat bin Rauf from Biofluid. This is from industry from Malaysia. This is good example letter for our university joining university with the industry, and also Prof. Dr. Sayekti from Universitas of Sebelas Maret. Yeah. Then, ladies and gentlemen, over the next 3 days, participants will engage in lecture discussions and a lot of activities, which is combining the understanding of science and practical experience. And then more importantly, we hope that these activities will spark new ideas, new research area, maybe innovations, maybe collaborations. Then, ladies and gentlemen, again, on behalf of Universitas 11 Maret, I welcome you all with warmly heart to this short course. Then, as requested by the committee, by saying Bismillahirrahmanirrahim, I hereby officially declare that the short course program 2026 with the title From Nature to Innovations, Exploring Sustainable Science in the Tropics is officially open. Again, thank you very much. Have a nice course and enjoy Solo. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Sigit Prastowo. Please remain at the front to present certificates to our distinguished speakers. We will also like to invite Professor Dr. Desi Suci Handayani, the Dean of Faculty of Mathematics and Natural Sciences, to present the souvenir to our speakers. Professor Dr. Taifo Mahmud, Professor Dr. Muhyusri bin Abrahman, Mr. Haizat bin Abrauf, Professor Dr. Sayekti Wahyuningsi, and Dr. Nurdiana Samsul Rizal. Please join us to the front for the certificate and souvenir presentation. Following the souvenir presentation, we will have a group photo session with our VIP guests, followed by a group photo session with all participants. For the participants, please come forward to join the group photo session. Ladies and gentlemen, thank you for joining the group photo session. We would like to We would like to express our gratitude to Professor Dr. Sigit Prastowo for his presence today. We wish you all the best for you continue with your activities, sir. Thank you. Ladies and gentlemen, we will now continue with the next agenda. Please take your seat. Ladies and gentlemen, we are now entering our first scientific session entitled Reason Advanced in Drug Discovery. The lecture will be delivered by Professor Dr. Taifo Mahmud from Oregon State University, USA. The session will be moderated by Apotheker Fitrawan Hernusa Pribadi, Master of Science. I invite the moderator to lead the session.
Oke, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Okay, Alhamdulillahirrahmanirrahim. Good morning, everyone. Okay, nice to meet you. Wonderful today. Okay, I will be want to see how motivated you are for today. Okay, when I say hello, you can reply hi. Okay, and when I say hi, you can reply hello. Okay, you can. Okay, can you? Okay, hello, hi, 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 hello, hi. Okay, everything you can say about that. Okay, thank you very much. Okay, the first, I would like to thank our honorable dean and vice dean and our for the committee for giving me to the wonderful opportunity to as a moderator for you all today because it's first time for me. So I'm so nervous today. So I'm apologize when I say okay, excuse me. Okay. Before we begin, okay, let me ask you. Okay, that's are you ready to learn something amazing for today? Okay, thank you very much because I glad you there when you motivated for today. Everything the material for our distinguished speaker will be impactful for you. Okay, awesome. Okay, today we are connected via Zoom with our distinguished outstanding speaker, Prof. Taifo Mahmud from Oregon State University. Hello, Professor.
Good morning.
Okay, good morning. Okay, I think in there at night, yeah, maybe, yeah. Okay, at night. Okay. So because in Surakarta and Oregon still 30 hours. Okay. The margin, yeah. Okay. Okay. So can you hear us clearly, Professor? Okay. Thank you very much. Okay. Excuse me for the computer. Can you show the portfolio? Okay. Thank you very much. Okay, let me to introduce our as distinguished speaker, Prof. Taifo Mahmud, PhD, currently serves a professor in the Department of Pharmaceutical Sciences at Oregon State University in the United States. Okay, he completed his undergraduate pharmacy and apotheker education at the University of North Sumatra, Indonesia. Okay, before earning his Master of Science and PhD degrees from Osaka University, Japan. As a researcher, he has published 142. Okay, excuse me. He has 140 peer-reviewed scientific papers. Okay, and achieved Google Scholar Edge Index of 45. Okay, more than wow. Okay, then I just one. Okay, it's like a Earth and the Moon. Yeah. Okay. He has significantly contributed to practical innovation by filing 10 patents application, including 6 US patent, and co-founding Gadusol Laboratories. Is that right, Professor? Okay. For your information, Gadusol is a chemical sunscreen, okay, that is found in the eggs of many fish species. Okay, so excuse me when I read an article that I found that uh, unique words. Okay, a fish N. Is that right? Okay, Professor, is a fish N. I think this sounds very familiar, yeah, to us Indonesian, yeah. So efficient, not but a fish N. That is or sunscreen from fish. Okay. Okay, throughout his distinction career, he has received prestigious honors like the Springer Nature Editor of Distinction Award and previously served as director of graduate programs at the College of Pharmacy, uh, Oregon State University. Okay, wonderful, Prof. Taifo. And with your background, okay, let's get right to it. Okay, you have 90 minutes, excuse me, Prof. Taifo, you have for presentation with the title is Recent Advance in Drug Discovery. Okay, and the floor is yours. Okay, excuse me, can you show the, yeah.
All right. Thank you, Pak Fitrawan, for the introduction. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. It is a really great pleasure to be here, through Zoom online. I would like to thank the organizing committee for giving me the opportunity to participate in this very important activities. So, as introduced, I was born in Indonesia and grew up in Indonesia, but I've been away from the country for more than 35 years now. But it's always nice to be able to come back from time to time, including through Zoom as well. So let me share my slide. All right. So, all right. So this international short course event or activities with a theme from nature to innovation, I think is very, very important, and it's close to my heart because I've been studying and working as a scientist on natural products, and then using more contemporary approaches to produce novel compounds through innovation, particularly using more recent approaches, including genetic and synthetic biology, and so on and so forth. So I was asked to talk about recent advances in drug discovery, which is, I think, is a good title for today. But before I start, I would like to um, share the place where I'm from. So Oregon State University in Corvallis, Oregon, the state of Oregon in the United States. This is the middle, the center of a campus. And this campus is pretty big, so even the College of Pharmacy is not shown here. My lab also is not shown here, but this is a center of the university. And we do have very interesting and unique buildings on campus. So it is recognized as the historical district or national historical district. So these buildings have been preserved for a long time. You can't really change them, at least the outside, but you can renovate the insides. All right, Oregon State University has been designated as a land grant, sea grant, space grant, and sun grant university. So in the US, only 2 or 3 institutions that have all these 4 designations, including Cornell University. So research in Oregon State University has been heavily on agriculture, marine science, as well as space and extraterrestrial research as well. So, so today, since we are talking about drug discovery, so I just want to remind you all the common therapeutic agents that we use currently. Mostly they are derived from natural products, right? That's one part of it. And then synthetic drugs, many compounds are synthesized chemically. And more recently, we have a lot of biologics as well, which are biological macromolecules like monoclonal antibodies, growth factor, and so on and so forth. But nevertheless, natural product has been historically very important for therapeutical agents. So natural products include herbal medicines or isolated natural products or natural product derivatives, right? And some natural products also being synthesized. So that even synthetic drugs, many of them really resemble natural products, or they use the natural product pharmacophores. So the sources of natural medicine, of course, as many of you know, um, the main one has been plants because we have used herbals and plants isolate or metabolites as drugs for centuries. And that includes Taxol, the anti-cancer drugs, morphine, quinines, and so on and so forth. But obviously, in addition to plants, microbe has been the prolific source of natural product as well. So fungi, bacteria, and other microbial organisms can produce so many different natural compounds or bioactive compounds. Those include penicillin, which is the first natural source of antibiotics, and doxorubicin, rifamycin, azithromycin or erythromycin, tetracycline, and so on and so forth. Animal also can produce certain compounds, mostly peptides, like teprotide or ziconotide, for example, or exenatide. So exenatide, perhaps many of you know, is used as anti-diabetic drugs because it's a GLP-1 receptor agonist, right? It's originally produced by the Gila monster, a reptile. It's called the molecule is called exendin 4. It's then based on that molecule, exenatide is synthesized through chemical synthesis to and be used as GLP-1 receptor agonist. And of course, nowadays, perhaps you have heard like Ozempic, for example, is widely used for to reduce, well, as a weight loss medication. And marine organism like spawns, tunicates, and even sea hair can also produce bioactive compound. These are a number of examples that have been used in clinics like trabectedin, dolastatin, and cytarabine. Many of them are used as anti-cancer drugs. So it's obviously a natural product continue to be an important source of new drug leads. If you look at all small molecule approved drugs from 1981 to 2019, about one third of them are either natural products, botanical or natural product derivatives. And another one third, there are synthetic compounds, but really based on natural products. Either the pharmacophores look like natural products or they are natural product mimics. Only one third of those compounds, small molecules that have been approved by the FDA, really purely synthetic compounds. So still, even today, natural products are the one of the most important source of medication or drugs. But unfortunately, traditional approach to natural product drug discovery, for example, like going out to collect sample and then isolate molecule and try to find new medication, has been more and more difficult nowadays because it's more and more difficult to find novel compound that way. So the rate of discovery of new compounds has declined significantly, and the isolation of known compounds has been the big problem, because it's we isolate the same compounds again and again, right? So obviously requires some innovative approach or different approach to try to find novel compounds, even though we know there are still a lot of molecules out there that we have not been able to isolate. So in the field of natural product drug discovery, because we try to include or use cutting edge technologies and creative approaches. Some people try to find new sources like organism from extreme and unique ecosystem, for example, or try to find new molecule from symbionts or microbiomes and so on and so forth. But you can also use cutting edge technology to speed up or to screen molecules in a very large scale, so that at least that would increase the possibility of finding new molecule. So you can use new tools, new techniques, or new screening system to do this. Another ways of doing this is by using creative approaches, by using biotechnology, using genome mining, genetic engineering, and so on. So today I would like to talk or discuss more about this biotechnology that has been used in natural product drug discovery. So my own lab here at Oregon State University have been using this technique for many years now. So this just to show you a number of publications that's representative for each of these technologies that we use. So number one is biosynthetic engineering, and then we will talk about chemoenzymatic approach, and then genome mining and synthetic biology. All right, so before I start, I just want to remind you, especially, if you don't have too much background in biochemistry, the central dogma of biochemistry, right? So when we talk about natural product, we are talking about natural compound or secondary metabolites. Those compounds are synthesized or biosynthesized in the cell of organism, whether it's plant or bacteria, fungi, and so on. The synthesis require multi-step reaction, just like when people try to synthesize a molecule using synthetic organic synthetic approach. So require many step reaction, and each reaction need to be catalyzed by something. So in the case of natural products, the reactions are catalyzed by proteins or enzymes. And of course, proteins, they are translated from messenger RNA, which are transcribed from DNAs or genes, right? So genes, they are in chromosome of the organism. So this kind of the basic concept of all the thing that we do. So when we do genetic engineering, basically we engineer the DNA or chromosome with the intention that we would engineer the system, the biosynthetic machinery, so that we can get different molecules. We can also use directly the enzyme in the chemoenzymatic approach to make different derivatives or different analogs of natural products. So in the general scheme of biosynthetic approach that we have used commonly is to establish the genetic information of the organism by genome sequencing. And then you can, depending on what approach you do, for example, you can do genetic engineering by, knocking out genes or swapping genes in the genome of the organism. So basically, you play around with the blueprints of that organism. And then the mutants that you produce, then you can optimize them in the culture to produce the compound that you expected. And that can be analyzed, by using LCMS and or other analytical tools to look for the metabolites that you are interested in. And then you can then isolate the compounds and structure elucidation. And finally, you can do bioassay or testing the activity of the molecule. Of course, this can be done in a different ways. For example, by doing bioassay guided isolation, for example, as well. So there are many different ways that you can do about to do this approach. So in the case of biosynthetic engineering, we can do gene knockout. We can knock out or disable specific genes in the genome that is responsible in making natural products. So that way we can uh, you know, maybe produce or um, control the organism to produce something different. You can also swap genes. The genes that you that is present in the in the organism maybe not the one that you want. You can swap with a different gene so that you can modify the molecule. More recently, of course, maybe you heard a lot about gene editing. You know, people can specifically change nucleic acid of the genes and then change the function or even knock out the activity of the genes. So one example here that I would like to discuss about is a biosynthetic engineering of pactamycin, which is a molecule, very potent molecule isolated from a bacteria, soil bacteria called Streptomyces pactum. So the molecule is very complex. This molecule was discovered by Upjohn Company many years ago. It has very good antibacterial activity, antifungal activity, antiviral activity, anti-cancer activity, but also very toxic to human cells. So it's very potent molecule, but it's not very useful because the toxicity is very, very high. As a chemist and pharmacist, I'm very interested in this molecule because of the structure. It's very complex, and with all the stereo centers, it's of course it's very difficult to synthesize this molecule chemically through synthetic approach. Even after 50 years after this compound was discovered, finally somebody was able to synthesize this molecule chemically, but of course the yield was very low. So we thought if we understand how this molecule is made in this bacteria and we can do genetic engineering or biosynthetic engineering, we may be able to modify this molecule without destroying the whole structures, right? So of course, again, we start by looking at the genes or the DNA and then express the proteins and then do the study to understand how this molecule is made. By knowing that, you know, you can use that knowledge to modify the molecule through biosynthetic engineering. So in general, molecules in nature are made through many steps, as I mentioned, but in a way like assembly line, like synthesis, right? Like making a car. So it's from a very small starting unit, and then it's being expanded step by step. So, you know, finally you make a very complex molecule that way. So example of this is like polyketide synthesis is enzyme that make polyketide compounds, or non-ribosomal peptide synthesis, and so on. So we spend a lot of time really to understand how this molecule, pactamycin, is made. So we clones all the genes and we do knockout, we heterologously express them, we characterize the enzymatic activity of each of these proteins. So the one that I wrote in a round or oval here, those are different proteins. You can imagine so many different proteins are involved in making this molecule from this small molecule called 3-aminobenzoic acid. So we thought going into detail just to give you some sense that you know, this biosynthetic pathway to pactamycin is very complex. And you know, this molecule is being attached to this protein is called acyl carrier proteins, and then being modified, extended, and then glycosylated, and then modification occur, and so on and so forth. And a lot of tailoring proteins here that modified this molecule to make the final products also present. By knocking out this gene or modifying this system, we are able to make different analogs of pactamycin. Here, just a number of example of them. We made more than that I show you here, but you can see the short version or the smaller version of them, and then the one with fluorine atom attached to this aromatic ring, and so on and so forth. So this is a cartoon that I made. So instead of going making pactamycin, you can block the pathway, and then so the pathway would take a detour to make different molecules. So some of them really show improved biological properties. As I mentioned to you just now, the parent compound pactamycin has very strong antibacterial activity, strong antimalarial activity, but very toxic. Just by removing this more functional group slightly, which is the thing that you cannot do through organic synthesis, because the CC bond here cannot be easily cleaved without if destroying the whole molecule. But we were able to specifically remove this methyl group and hydroxy group to make this pendant methyl. And the product here completely has no antibacterial activity, very less toxic, and still retain very good antimalarial activity. So just by modifying this molecule slightly here and there, we were able to make a much better compound. In this case, you know, against malarial parasite. All right, so that's one approach that you can use to make a new molecule, or in the case of drug discovery. But you can also use chemoenzymatic approach, basically combining traditional chemical method with biotransformation or using enzyme to build complex molecule. Again, if you study the biosynthesis of natural products, you will find many different enzymes. Some of them can be useful for this kind of activity or approach. So this will you will be able to bypass, long and tedious laboratory steps just by combining enzymology in your synthetic approach as well. So as I mentioned just now, natural products are synthesized in a kind of in the assembly line like synthesis. So here the example is conglobatin. This is a dimeric macrolide dialactone. This is a very strong anti-cancer agent as well. It's produced by Streptomyces, also by Streptomyces pactum. These compounds inhibit cell proliferation in HER2 plus breast cancer cell line. So because this is a dimeric compound, of course, this molecule is derived from two monomers. So two monomers here actually is produced through this whole kind of different enzyme or proteins from this formyl glycine, and then being extended, to make these two monomers. And then the thioesterase domain here is the one that responsible for dimerization reaction to make this molecule. So remember, this thioesterase is the one that make this dimer. But interestingly, we found that in addition to making this dimer, this thioesterase also can make a different molecule, basically incorporating different natural products into the reaction and coupling this molecule with the monomer of conglobatin. So we were able to isolate this type of molecule in the producing bacteria. So more interestingly, because NFAT133 here is derived from a completely different biosynthetic pathway. So in this case, two different biosynthetic pathway merged together or combined together by this thioesterase domain to make completely new molecules. So we confirmed this by making the monomer. So we took the dimers, chromobatin, and hydrolyzed it to make a monomer and attach these small arms. Re really is resembling the phosphopantethenial arm of these acyl carrier proteins, because this monomer usually attached to the acyl carrier protein. So making this molecule is quite difficult, so we just make a small or a short version of it. And that's, did the trick here, because the enzyme was able to take this molecule and then catalyze the coupling reaction with MPET 133 to make this hybrid molecule. But interestingly, when we replace this NFET 133 with other nucleophile like this molecule, all of them has hydroxy group or amino group, we are able to make analogs of this compound. So with different side chains such as this attached to this R position, right? So using chemoenzymatic reaction, you really can make new analogs of natural products. Similar thing, we have done the same thing with pactamycin as well, using this enzyme called PTMR, which is in the real biosynthetic pathway in responsible for the transfer of this 6-methyl salicylyl unit to this hydroxy group to make pactamycin. So again, instead of making the real or natural molecule, we just make this short version of it just to resemble these arms. And this molecule was able, well, the enzyme was able to use this molecule to make pactamycin. Similarly, we can replace this 6-methyl salicylyl group with many different acyl group here, and we were able to make analogs of pactamycin containing all different side chains, right? So there's another, really the beauty of this approach, really, you can do all this thing in the test tube or in the append of tubes to make different natural products. All right, so the next approach is genome mining. So basically, from the name, you may guess, basically we mined the genomic information of one organism to see, what kind of molecule the organism can make, and then we try to chase them or to isolate the active molecule. So it's really exploiting genomic information for the discovery or the biosynthetic pathway of natural products and their products. Well, of course, it's require computational technology and bioinformatics tool, but all those tools are easily available nowadays. You can use many different tools available online. But the whole idea is that even though we have been able to isolate many compounds from organism, particularly like soil bacteria, apparently there are so many compounds or biosynthetic pathway that we haven't touched so far. Just for example, if you take 1 gram of soil bacteria, in that 1 gram of if you take 1 gram of soil, in that 1 gram of soil, actually, there are about 10 to the 10s bacteria in there. Maybe about 4 to the 4 times 10 to the third and 5 times 10 to the fourth species in there. So many of them. But so far we have only been able to culture about 1% of those bacteria in the lab. So if many of those bacteria, we cannot really culture them in the lab because, well, we don't know how. And many of them maybe require a very specific condition or maybe they are anaerobics and so on and so forth. So only 1% that are culturable. And even from that 1%, you can imagine how many, there's so many already. If you take one strain only, in this one strain, presumably you have more than 30, between 20 to 40 biosynthetic gene cluster, meaning cluster of genes in the genomes of that strains that can make natural products. Meaning 30 biosynthetic gene cluster means this organism can make 30 different natural products or classes of natural products. But the reality is, if you look at all the strains that have been studied quite extensively, for example, Polyspora erythrea here, it has, well, the genome size is about 8.2 megabase pair, and it has about 27 BGC, meaning biosynthetic gene cluster. But so far, only 5 of them have been known in terms of the product, the natural products. Like Streptomyces spectrum has 31 biosynthetic gene cluster, but only 4 different classes of compound have been isolated from this particular strain. So like 27 other biosynthetic gene cluster are still unknown or cryptic, or we don't really know what they make, right? So that's the reason why this genome mining approach will perhaps give us the opportunity to look for them or to induce their production so that we can get new molecule from the same organism that has been studied for many, many years. So if you look at the HPLC trace here, so this is from Streptomyces spectrum that has 31 putative biosynthetic gene cluster, only 4 different group of compound, NFET133, this is pactamycin, this is congloberdin here, and this is pyrisidins, right? So the other 27 group of molecules, they are not here, they are nowhere to find. All right, so um, obviously we need to find a way, how to induce or how to encourage the bacteria to produce them. Unfortunately, under regular laboratory conditions, sometimes they just produce very few natural products. So apparently all this production of natural products, really they are regulated tightly by global and pathway specific regulation. So it's not randomly being produced, right? There are regulators and elicitors and so on that really control the production of those natural products. So by studying regulators or regulation of natural product biosynthesis also um, may lead us to the ability to produce novel compounds. So that's part of the work that my lab has been working on as well, but I don't have time to talk about it right now, maybe in another time. So, but to turn on silent biosynthetic gene cluster, or to increase secondary metabolite production, there are several methods that have been used widely in the field. That can be specific method or non-specific method. Of course, specific method really requires some more knowledge and technical skill or needs to make them to happen, like doing engineering of self-resistant or heterologous expression, combinatorial biosynthesis, and so on. But many people use very easy non-specific method, for example, by manipulating media or changing the media that are used, or inducing stress response, or do random mutagenesis, like many pharmaceutical company did many years ago, or use elicitor, for example, or co-cultures, because those bacteria in nature, they are not living alone, right? They live with many other organisms or microbes. So you can maybe try to co-culture them and see if that would induce the production of secondary metabolites. So one technique, well, recently our lab found a specific small molecules that function as a regulator or as elicitors. So using that molecule, we were able to induce the production of certain natural product or to induce the yield of the production of that natural products. So for example, by adding this molecule into Streptomyces silicolor, this is a model organism that produce this blue pigment called actinorhodin. So by adding this elicitor into the culture of Streptomyces silicolor, we can induce the production of actinorhodin very early at 72 hours after inoculation. Without feeding or without elicitor, you can see the compounds only being produced perhaps after 96 or 120 hours. And the yield also completely different. Not only this molecule, well, we use this actinorhodin as an example because it's very nice, colorful blue pigments, right? So very easy to see the effect of this elicitation. But this elicitor also induce the production or regulate the production of many other secondary metabolites in these strains. You can also do genetic engineering, for example, by knocking out certain genes or certain regulators that down-regulate the production of natural products. And then you will get mutants that produce different type of compound. You can see this is the wild type, and then this the mutant. And then these are the HPLC chromatogram to show these are three mutants and three wild type and three mutants. You can see the secondary metabolites that are produced by the mutants are quite different from those produced by the wild type. So using this technique also you can induce the production of natural products that have not been observed before. So, and the same way you can also regulate antibiotic production. For example, this is the wild type, the pink one. So this cluster of molecule, this is the molecular network of the metabolomes of this strain. This pink one, this is the cluster of compounds that are only produced by the wild type. But when you knock out certain genes, the mutant is no longer produce this molecule, but produce something else. So the green one only produced by the mutant strain. So you can really change the metabolite production of certain organism by doing this genetic engineering, or you can improve the production of antibiotics. So as you can see here, the red one here is the mutant. The concentration of antibiotics being produced is significantly higher than the wild type, right? So all this kind of technique really can induce production of even minor metabolites, the one that typically we don't see, but after genetic engineering, you can increase the production of those molecules. So on the other hand, you can also completely remove the production of the compounds that you already know that you don't need anymore. You can make a mutant that's no longer produce those major compounds. Then you are left with the molecule, the one that you only want, So that's really improve the purification process, and also you can get small analogs that typically you don't see as well in the wild type. So in doing this way, we were able to isolate different analogs of conglobatin with different side chain, for example, here and there. And some of them really have better biological activity compared to the natural products, the original natural products. So lastly, I would like to talk about synthetic biology, which is a field of science that involves redesigning the organism for useful purposes, depending on what you need, right? By engineering them to have new abilities, right? So this synthetic biology has been used in many different fields, including in the field of energy. For example, people generate algae to make biodiesel or biofuel, for example, by modifying the organism genetically, or creating a new completely new organism with new capabilities, for example. In our case, one other aspect of our research really looking for natural sunscreen, and apparently marine um, organism or good resources or good sources of natural sunscreen, because as you know, marine environment is very harsh with sunlight and UV radiation all days. Many of these organisms, they are capable of making sunscreen to protect them, or perhaps they can take the natural sunscreen from their diets or from symbionts, like microorganisms, cyanobacteria, blue-green algae, or diatoms. They can make a molecule called mycosporin-like amino acid like this, which can protect them against UVA radiation. Algae also can produce mycosporin-like amino acid, like red algae or brown algae. And then coral, for example, can make mycosporin-like amino acid by themselves, some of them, but some of them also really rely on their symbionts. Marine invertebrate as well usually take those molecules from their diets or their symbionts. And fish can get MAA or mycosporin-like amino acid from their diets. But we found that fish can also produce another sunscreen compound, gadusol, de novo by themselves. Just now, the moderator introduced you a bit about gadusol, this sunscreen molecule that can protect the fish against UVB. So these are really important. The reason why we are interested in this, because as you know, the many sunscreen product that we use currently, they are really contain synthetic sunscreen like oxybenzone, octinoxate, and so on. Those synthetic sunscreen, they can damage coral ecosystem. They cause coral bleaching. They kill coral embryos. So many places have banned the use of those sunscreen in the area. And then more recently, we found, or people found, that this molecule also being absorbed systemically, get into your body, even though we don't really know what the health risk of that. And zinc oxide and titanium dioxide, those are mineral sunscreen, but as you know, the formulation is quite difficult, and then if too much, then it's get white, then your face getting pretty white because of the zinc oxide, right? So because of that, we set out to develop a system to produce gadusol, but of course not just directly taking the molecule from fish, right? Even though you can get the molecule from the fish eggs, but instead we use synthetic biology approach by taking the genes that are responsible to make this molecule from the fish. So we take genes from fish that can make this molecule, and we transfer them into yeast, Saccharomyces cerevisiae, or put them in a bacteria, and we were able to make the molecule that way. So this yeast, you can culture them, you can put them in the fermenters, or bacteria can make the same molecule, the same compounds, gadusol, that absorb UV light at 294 nanometer. So it's a natural sunscreen against UVB. And we were able to increase the production yield as well, 1.5 gram per liter of culture. And this experiment show that these are yeast actually grow on the plates. If you add gadusol, then well, you expose this yeast with UV lights, with different doses here. So, but the one without gadusol, usually they die, but the one with gadusol, they survive of this UV exposure of this yeast, right? Um, but gadusol only protect us from UVB. So if we want to make molecule that can protect us with against UVA, then we need to extend this molecule a little bit longer. So what we did then, we took one gene from a fish, one gene from a soil bacterium, and two genes from plant pathogen. Of course, we have, studied a lot about these genes, and we know their function. And we put together all these different genes from different organisms together in one cassette, and then introduce them into this bacteria called Streptomyces silicolor. We were able to make this completely novel molecule that we call gadosporin, that can absorb UV light at 340 nanometer. That's protect us from the UVA. So now we have two different molecule that can cover both UVA and UVB radiation. So yeah, so as mentioned just now, so this technology has been licensed. We studied a company called Gadusol Labs, but then that's company being purchased by another one that they obtained the license, and they are now working on developing this type of molecule as a novel natural derived sunscreen products. All right, with that, I would like to summarize. So I hope I was able to clearly explain to you that cutting-edge technology and creative approaches have changed the way how drug discovery and development research is conducted. Biotechnology, obviously, the future, well, current and future, I guess, that will continue to play a major role in drug discovery and development. And some example like biosynthetic pathway engineering, chemoenzymatic approaches, genome mining, and synthetic biology really have opened new opportunity for discovering and developing new drugs. And using this technology, as I have shown you, many, many novel bioactive compounds can be produced. With that, I would like to thank you know, people who are involved in this study, and then thank you all very much for your kind attention. And I'm happy to answer any question that you may have. Thank you.
Yeah. Okay, thank you. Big applause for Prof. Taiwo. Okay, thank you very much, Prof. Taiwo, for the amazing presentation and I think valuable insight. Yeah. Okay, now we open the question session. Okay, for anyone will be the give a question, can you raise hand and wait for the microphone? Please help committee for the giving the microphone. I think you can say your name and your institution before. Okay. I think in yes, excuse me. Yeah, in my left hand.
Assalamu alaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Basically, my name is Muhammad Umar. I'm from Nigeria, bachelor student of biology from the Department of Biology, Faculty of Mathematics and Natural Science, Universitas Sebelas Maret. Thank you, sir, for your time and your guidance. And we hope that the knowledge that you give us is going to give an importance to us, inshallah. Okay, sir. You have said something previously. Definitely have some question. I have two questions with me here. The first question is that why are natural products are still important in modern drug discovery? The first question. What the second question is that what's different between natural product and synthetic drugs? So I think the question are clear. Thank you very much.
All right. So the question is, why are natural products are more or are better than synthetic compounds, right?
No, the question is that why natural products are still important in modern drug discovery? Yeah, this is the first
Right.
question.
All right. That's correct. All right. Um, so about 30 years ago, well, yeah, I think some around, yeah, 30, 40 years ago, a lot of pharmaceutical company, big pharma, decided not to work on natural products anymore, um, because they found a new ideas with the advance of technology, with all this high throughput screenings. They thought they can just make millions of compounds synthetically, right? And screen them for active molecules. So because natural products, obviously, as I mentioned, it is more and more difficult now to find novel natural products, right? So the approach has become more expensive and time consuming and labor extensive. So the big pharma decided to abandon natural products and then just make compounds synthetically and then screen them. But the result of that, well, is a complete failures because they didn't find really a good molecules that way. Apparently, um, when you try to synthesize molecules synthetically, even though you make many, many molecules, but the diversity of those molecules is very limited, the range of diversity. On the other hand, natural product is very diverse. All right. Apparently, nature has evolved for million years of for million years really to selectively making many different molecule with very wide diversity and with very very good biological activities. So for that reason, even until today, even modern time, um, natural products still um, are still important for drug discovery, or at least still a good example, or give us inspiration, even though if we want to synthesize molecule, but the inspiration really coming from the chemical structure of natural products. All right. For that reason, many drugs now we have these derivatives of natural products. And to be honest, nature made those compounds. I don't believe they make those compounds to treat our disease, right? So they made those compounds for their own purposes. Sometimes they use the compound for communication, signaling, or they use the compound for protecting their niche, for competition. Um, but then, because those compounds are biologically active, then we use them as drug. But if you pay attention, not that many, um, well, many natural products in the natural form cannot be used directly. In many cases, we need to do modification to improve their uh, ADME, the absorption, the metabolism, and so on. So to improve the pharmacokinetics, even pharmacodynamics of the molecule. For example, the natural penicillin is only active against gram-positive bacteria, and many gram-positive bacteria are already resistant to natural penicillins, right? Uh, but then uh, what we do really, we modified the natural penicillin or penicillin G, right? We make like ampicillin, amoxicillins. Those are not natural products. Those are derivatives of penicillin, chemically synthesized. Like azithromycin is not natural products. The real natural product is erythromycin, but erythromycin is very bitter, and the absorption in the gut is not good and very sensitive to acid. That's why we make azithromycin or other derivatives of erythromycin. The same thing with tetracycline. The natural product chlorotetracycline is no longer used or very limitedly used. But what we use now is doxycycline, right? Because doxycycline has better pharmacokinetics properties. All right. So again, although natural products are sometimes cannot be used directly, they can be used as to inspire us to make a better drugs. I hope that answered your question.
Yeah, thank you very much. I think it's clearly done well, Prof. Taifo. Okay. Okay, excuse me, the number two. Okay, excuse me, Prof. Uh, please, the committee. The second question, Prof. Taifo.
Sure.
Okay.
Thank you very much, Professor Taifu. I was from Faculty of Mathematics. 4 years ago, I invited you in Sebelas Maret by online as well, yeah, because I'm secretary of academic Senate academic. I still remember. Okay, thank you.
Yes, yes.
Okay, my question regarding about a long discussion genetic modified food and genetic modified organism. There is some assumption for the long term can cause cancer. This is the first question. And the second question regarding about, the genetic engineering. There are three type gene, what is gene knockout, and then gene, what we got, editing, and also the other one I forgot. There is the type of the gene mechanism. So which one is the best one to produce some what is the new what we call it, the new product for, especially to produce, to increase the production of our food. This one, which one the best one? Thank you very much, Professor Taibo.
Thank you. That's a very important question about GMOs. Of course, you know, I think it's really case by case. Well, in the case of food, GMO, for food productions, um, I know, many places concerned about GMOs, but unfortunately or fortunately in the US, most of those foods, they are GMOs pretty much, because even the beans and everything, and then, well, chemically, even though the gene has been modified, they are all nucleic acid. Scientifically, they are all nucleic acids, and then, so when you eat them, of course you will digest them completely, so it doesn't matter really the genetic makeup of those, but of course, some people concerned about that, because it's food, then you eat everything, the genes include in those material that you eat. But in the case of drug discovery, even though we do genetic engineering, but it's just to make the molecule, the compounds. At the end of the day, what you isolate is the chemical molecules, right? And then pure, that's the one that you use as medicine, as drug. So purified chemical entities. In that case, there is no more genes involved in there, right? Even though the way how you make it, perhaps you use genetic engineering, but the end products is a purified compounds, does not contains any genes. So it's a slightly different context here. But in the case of food, yeah, if you eat corn, right, or if you eat rice, so the genes that the engineered genes also in the food, the one that you eat. So if you're afraid of that effect, then yeah, that's a reasonable concern. Even though, like I said, once those those are still nucleic acid when you eat, and it will be digested, and they all will be the same materials. So genetic engineering just changing the nucleic acid, but still let's contain ATCG. Just the sequence may be slightly different. All right. And about the genetic engineering technique, the uh, gene knockout, and then gene swapping, and then the gene editing, those are different technique. I cannot say which one better. Really depend on what your purpose is. What you want to do about it. For example, if you do gene swapping, um, yeah, you cannot do that by using say gene knockout. So there's two different purposes. And then the gene editing, CRISPR Cas9, for example, has been used widely. Of course, people get Nobel Prize for that, and those technique works very well in yeast and human cells, but in bacteria, at least in our hand, it is more difficult. So we decided to do more traditional way of genetic engineering rather than using CRISPR-Cas9 system, for example. So I hope that's answered the question.
Okay, thank you very much. I think it's clearly Prof. Arihandono. Okay, I think because we have just a limited time, I think just one again participant. Okay, okay, excuse me. Oh, maybe two, yeah. Okay, in my right before. Okay, yeah. Excuse me, the microphone please come with. Excuse me, Prof. Hari. Okay, have done. Okay, thank you.
Hi. Assalamualaikum, Prof. Okay, my name is
Waalaikumsalam
Nabila
warahmatullahi.
and I am from UITM Shah Alam, Malaysia. Okay, I want to ask you three questions. Okay, the first one, what is your opinion in combining natural products in a real product that can be consumed by consumer lah? Okay, the second one, okay, I just want to ask about the 1% of soil microbes that you mentioned earlier in your slide. I just wondering, why is it sounds impossible to be culture? Is that because of what is the limitation? Is that because of the media, or the condition is under study, or anything else? So that is the second question. And then the third question, I'm interested in the coral bleaching study. Okay, we are facing coral bleaching. Is that the work you have done? Is one of the effort to improve the condition of our sea or the coral? And is it already being implemented in the real life? Thank you so much, Prof.
All right, thank you. So the first question, when you say incorporating natural product into a product, are you talking about herbal or is purified natural products?
Yes, herbal.
Herbal. So yeah, so my opinion is quite clear. As long as you have enough, pharmacological data and toxicology and clinical data to support that, that's okay. But in many cases, because it's called herbal, so the regulation is very light. And in fact, there are many herbal medicine actually contains very nasty chemicals. Like tongkat ali, for example, right? It's being used in Malaysia maybe a lot. It's contain quasinoids compound that has, like can cause liver toxicity. But of course, when people take it, occasionally, maybe they don't really realize that. All right. So yeah. So as long as you have enough clinical data to support, I think that's fine. And the second question is about 1%. So yeah, so many people call it, the 99% is unculturable, right? But that's kind of a very, you know, very well, unspecific response. Unculturable, what does that mean, right? Perhaps the real meanings really, we are not smart enough to find out, what is the best condition for those bacteria to grow. Because of course, natural conditions sometimes very unique. The bacteria maybe cannot grow by itself, require some chemicals from a different bacteria. They are doing like symbiotic life, right? So yeah, so there are many factors. And then of course, in the lab, we pretty much arbitrarily, we just decide what media we are going to use. We never ask the bacteria what media they want, right? Of course, we cannot ask them. So we try our best. We try different media, and then some of them work, some of them not. But apparently, many bacteria are still not. They don't grow. The third question or the answer is, well, I'm not an oceanography, even though we discovered that those compounds can protect coral. But the reason why coral bleaching really caused by many factors, right? Environmental factors and also chemicals and a lot of toxin that we put into the sea, the ocean, and those are the one that kill corals. So this is, of course, is not an easy task. Obviously, that's why some places ban the use of certain sunscreen products so that we don't, dam more chemicals into the ocean. But as you can see, there are a lot of plastic, a lot of other stuff in the ocean now. So yeah, so it's a bigger problem to handle, I guess. Thank you.
Okay, thank you very much. Okay, the last question. Okay, excuse me, Prof. Taifo. The last question,
Uh-huh.
maybe. Okay.
Yeah, that's fine.
Okay, excuse me. Okay.
Go ahead.
Yeah, okay.
Thanks for the time having me. My name is Nurul Kusuma Devi from Biology Education Department, Universitas PGRI Madiun. Thank you, Prof, for the insightful, excellent, and clear presentation. We know that synthetic biology of natural product has many advantages especially in terms of efficiency and also sustainability. But is there any disadvantage or weakness of synthetic synthetic biology of natural product compared to sourcing from nature? Thank you.
Well, thank you very much. A very good question. Of course, every factor always, every approach has a different benefit and risk. And synthetic biology, one of the problem really, sometimes, the regulation, right? So even though we are able to put them together sometime, because the genes are, in a different system, is called heterologous system. So sometime the yield is low, for example. But all those things can be fixed, or you can study it and improve the production yields. And some organism maybe they do not produce the precursor that you need to make the molecules. As I showed just now, to make one big molecule require many different building blocks, like 3-aminobenzoic acid or acetic acid, or, many different components. And some hosts or some heterologous hosts, they do not produce those, so you cannot make the compounds. So meaning if you want to use that host, then you need to introduce other set of genes that can make the building blocks, right? So yeah, many aspects need to be considered when you decide where to or what organism to use to in this synthetic biology approach, right? So yeah, so those are more technical thing, and yeah, sometime the natural source is just too difficult to get, right? Like coral, for example, or even fish, like gadusol, it's impossible to collect so many fish, to get the molecule from the natural resources. So the one way of doing it really to move the genes into a organism that you can grow in the lab or in the factory. The same thing people make artemisinin, for example, artemisinic acid, that's anti-malarial agents. So naturally you get the molecule from the plants, right? Artemisia annua. But then now people are able to make the molecule in yeast. The same thing with morphine as well. So yeah, so this synthetic biology approach is kind of a rather new, but I think it's a future of many things. You can make many different compound that ways.
Okay, thank you very much, Prof. Taifo Mahmud. Okay, I think it's clearly done answering, yeah? Okay. Okay, I think because we have just a limit time, so I maybe in the closing session. Okay, let me to resume our topics. Excuse me, Prof. Taifo. Recent advances in drug discovery show that natural products remain a valuable source of new medicines, supported by modern biotechnological approach. Techniques such as biosynthetic engineering, chemoenzymatic synthesis, genome mining, and synthetic biology enable the discovery and development of novel bioactive compounds. Okay, thank you very much, Prof. Taifo Mahmud, for your valuable insights. Okay, I can give the big applause. Okay, thank you very much,
Thank
and thank
you.
you for the audience for the great question. Okay, and thank you for everyone. I apologize if I do mistake as a moderator, and have a good day. Okay, and the time given back to the master of ceremony. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullah.
Thank you, Professor Taifo Mahmud and Mr. Fitrawan. Please return to your seats. We now move to our second session entitled Natural Solar Cells, presented by Professor Dr. Muhyusri bin Abdurrahman from University Kebangsaan Malaysia. The session will be moderated by Dr. Lutfia Kurnia Permatahati, Master of Science. I invite the moderator to take over the session.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Good morning, all participants, distinguished speakers, and all lecturers of Universitas Sebelas Maret. Thank you so much for the organizing committee for giving me the opportunity to serve as moderator in this session. Let me introduce myself. My name is Lutfia Kurnia Permatahati from Basic Department. Maybe some of you already know me. since I have been communicating with you through WhatsApp. Yeah, that's me. Actually, I also never seem like Mr. Fitriawan before, but try to calm down. Yeah. Okay, this session, we are honored to have Prof. Yusri as a speaker, and he will deliver lectures about the SSE, dye-sensitized solar cell. But before I officially invite him to come forward, let me introduce him first. Maybe the committee, can you show up the CV of Prof. Yusri? Yeah. Okay. Professor Dr. Muhammad Yusri Abdul Rahman is a professor at the Institute of Microengineering and Nanoelectronics, IMEN, University Kebangsaan Malaysia. His research focus on dye-sensitized solar cell, energy materials, and nanomaterials. He earned his PhD in physics from University Kebangsaan Malaysia at 2005, and he obtained his bachelor and master degree at University Science Malaysia. He has published more than 100 papers in international journals. He also received several awards, including Outstanding Reviewer Awards from Elsevier and etc. Okay, without further ado, please welcome Prof. Yusri. Please give applause to Prof. Yusri. Before we start the lectures, I will remind you again that this session will be similar with previous session. Our speaker will deliver a presentation for about 1 hour and 30 minutes, and after that, followed by question and answer session for about 20 minutes. So I ask you to take note during the presentation and prepare your question. So at the end of the session, you can ask directly to Prof. Yusri. Yeah. Okay, Prof. Yusri, are you ready? Okay, Prof. Yusri, the floor is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and good morning to everyone. Okay. So the topic of my talk is about dye-sensor solar cell, fundamental and current trend. Okay. Okay. At first, we look at the outline of the talk. I will start with introduction to UKM, introduction to IMEN, okay, Institute of Microengineering and Nanoelectronics, then move to solar cell, several aspects of solar cell, focusing on DSSC, which is the full name of dye-sensor solar cell and its charge transport mechanism. Okay, the next process that I will discuss with the audience is the important or critical process in the device, that is interfacial charge transfer. Then I move to the component of each device. Firstly, is photoanode, sensitizer of the SSC, or which also called dye. The next component is counter electrode. Last component is electrolyte. Okay. Then move to the performance study of the device. Okay. There are several performance studies. We start with current voltage, electrical impedance spectroscopy to study the charge transfer, incident photon current efficiency, cyclic photometry, polarization interval, DSC with various counterdiode that I have studied so far, and I will end the talk with future work on the dye-sensor solar cell. Okay, at first, I would like to introduce my university. The name of the university is University Kebangsaan Malaysia. It has 4 campuses. The main campus is located at Bangi, okay, with the address shown on the screen. The second faculty is Faculty of Medicine, located at Cheras. The third branch is Faculty of Health in Kuala Lumpur. And the last one It's in overseas, in Qatar. Okay. So it is also called UKM Qatar branch. Okay. So UKM is one of the 5 research university in Malaysia. Okay. It is ranked at number 2 in the country and ranked in 138 in the world. Okay. So rank 138 according to QS World University Ranking in 2025. The top university in Malaysia is University of Malaya, ranked at number 60 in the world. So currently, UKM has more than 25,000 students with almost 15K undergraduate students. Okay. And some of them are masters and PhD students. Okay. So currently, UKM has 23209 staff, including academic staff, management staff, and support staff. Okay. The current photo shows the main campus, located at Bangi. Okay. So my office is in the main campus. Okay. The second campus is Trust campus, focusing on medicine. Okay. The third one is located at the city of Malaysia, Kuala Lumpur. Okay. Okay. The last one is so wonderful campus. It has one building only, okay. Located at very rich country in the world, by the name of Qatar. Okay. Next, I would like to introduce my institute by the name of Institute of Microengineering and Nanoelectronics. Okay. And the short form is IMEN. Okay. So UKM has 13 faculties and 13 institutes. Yeah. And IMEN is one of them. Okay. Institute focus on research. Okay. To become a research university, the university must have research institute. Okay. It cannot be all faculty. Okay. Next is 5 research group in IMEN. Okay. The first is MEM, second nanophotonic, third microelectronic. The fourth one is organic hybrid flexible electronic. Okay. And the last one is nanoelectronic system. Okay. My group is organic hybrid flexible electronic, okay, with the short form of OFFE. Okay, now we come to the content of the talk, solar cell. Okay, what is solar cell? Okay, solar cell is a device that converts light energy into electrical energy. Okay. Solar cell have 4 categories. First category is silicon solar cell. Silicon solar cell have 2 types, monocrystalline and polycrystalline. The highest efficiency of monocrystalline silicon solar cell is 26.8%. The one made of polycrystalline is 24.4%. But the disadvantage of silicon solar cell is the cost is very high. Okay. The way to process a silicon solar cell is expensive, and the material making up the cell is also expensive. Okay. So nowadays, silicon solar cell is not suitable anymore. And researchers in this field move to third type of solar cell, okay, and one of them is dye-sensitive solar cell. Okay, the second generation of solar cell is amorphous silicon, thin film solar cell made of cadmium telluride, copper, indium, gallium, diselenide. Okay. If we compare the if we compare the efficiency of second generation with the first generation, we found that the efficiency of the second generation is slightly lower than that of first generation, but the cost of second generation solar cell is lower than that of first generation. So we have the pro and con here. High performance, lower cost. Higher performance, lower cost. Yeah. The third generation has three types: dye-sensitive solar cell, perovskite solar cell, organic solar cell, and another one is quantum, not three, four. Okay. If we compare the efficiency, DSSC has the lowest efficiency, okay, with the highest efficiency of 14% using single dye sensitizer. But when we compare the cost, DSSC has the lowest cost among all types of solar cell in the third generation. Okay? So we have pro and con here. Okay? And the last type, tandem or hybrid solar cell. Okay, we look at the efficiency produced by tandem solar cell. Tandem means we combine two types of solar cell at least. Okay. Okay, for the first case, perovskite is tandem with silicon, producing 31.46%. It's very high. Okay. The second, perovskite CIGS tandem, okay, produce 29.7%, but the cost is high, and to manufacture is complicated. Okay, so hybrid solar cell and tandem should be manufactured by industry, not academia. Okay, academia focus on fundamental aspect of solar cell. Okay, so my responsibility is to publish in world journal, not to make product. Okay, this is not my work. Okay, so it's challenging if academia do all the things. Okay, so academia focus on fundamental. Produce or publish paper in journal, international journal. Okay, when we talk about solar cell, okay, we must ask ourselves, okay, what are the parameters of solar cell? There are four basic parameters to characterize the performance of the device. Okay. There are four parameters. First, power conversion efficiency, okay, with the symbol of mu. The second is short circuit current density, okay, with the symbol of GSC. The third one is open circuit voltage, VOC. And the last one is a fill factor. Okay. The most important parameter is power conversion efficiency. Okay, how to get power conversion efficiency? It is easy. So we multiply GSC by VOC by fill factor, and then we get power conversion efficiency. It's easy to get power conversion efficiency. Okay. So I choose several solar cell, several organic solar cell, okay, made of four materials: P3HT, PCPDTBT, PCDTBT, PTAA. Okay, please ignore aurum. Aurum is not organic material. Okay, aurum is counter-electrode material. Okay, according to the JV curve, okay, the solar cell made of PTAA produce the highest efficiency. This is because this device has the highest GSC and VOC. Okay. Okay, now move to dye-sensitive solar cell. Okay, the type of the solar cell that I work on together with my S2 and S3 student. Okay. Who invented this device? Okay, the device was invented by Professor Michael Gretzel in Switzerland in 1985, long time ago, about 41 years ago. Okay. And Michael Gretzel got the efficiency slightly above 2%, but published in the famous or prestige journal in the world by the name of Nature. Okay. But even though efficiency is only 2%, but Michael Gretzel invented the device. Okay. So the device has four components. TiO2, which is photoanode. Photoanode serves as electron acceptor. Okay. On the left of TiO2 is dye sensitizer. Okay, the name of the dye is not mentioned here. Okay. On the left of dye is electrolyte. D plus and D are the species in the electrolyte. Okay. The species are in the form of ion. And the counter electrode is made of platinum. Okay. Platinum is very famous material for DSSC because it has the highest catalytic activity and the most stable counter electrode material. Okay, the characteristic features of DSSC, the fabrication process or assembly process are straightforward and low cost. All the material making up the device has low toxicity and semi-transparent. And the device is colorful and beautiful design. Okay, the color of the device based on the color of dye. Different dye has different color. And the maximum efficiency nowadays for this device is 14.1%. Okay. So the device uses single dye. Okay, look at the structure of the SSC. So we assume light coming from bottom. Okay. Okay, the white dot represent TiO2 particle. The white dot represent TiO2 particle. It is coated with dye molecule. Okay. The device has a redox couple triiodide with the symbol of I3 negative and triiodide I negative. Okay. Above electrolyte is platinum colored in gray. Yeah. And the photoanode made of TiO2 and platinum are prepared on the substrate, the TCO, transparent conducting oxide substrate. So there are two types of substrate, indium tin oxide and indium tin oxide and fluorine dope tin oxide. Okay, the figure on the left shows the solar panel made of DSSC. Okay, normally we see solar panel made of silicon. Okay. But the current photograph shows the solar panel made of DSSC. Okay, we need to know a bit about the charge transport mechanism in the device. Okay, the first process is light absorption by photosensitizer. And then the electrons are excited from HOMO of sensitizer to LUMO of sensitizer. Okay. So sorry, this is quite theoretical, okay, but I need to explain a bit about the process that takes place in the device. Then the injected electron from the LUMO is injected to the conduction band of TiO2. Then the electron finally reaches the counter electrode through an external circuit. Okay. So the dye molecule at ground state, okay, is transformed into dye molecule at excited state. Okay, the symbol of S become S asterisk. S with asterisk is excited dye molecule. Then excited dye molecule transfer the electron to conduction band of TiO2. So S asterisk become S plus. So S plus is oxidized dye molecule. And oxidized dye molecule is transformed back to S. Okay? So S is dye molecule at HOMO. Okay? So the process must be recycled. And the recycle process also take place in electrolyte. Okay. For which triiodide is transformed to iodide. Yeah. So triiodide take 2 electrons from counter electrode to transform to 3I negative. Okay. Upon light illumination, the process is recycled. Okay, the schematic diagram shows all the processes take place in the device. Okay. So I'm not going to explain in detail because it's too theoretical. Okay. Okay, now I would like to share with you each component of the device one by one. I start with photoanode. The famous material for photoanode is TiO2. And TiO2 must be in nanometer size in order to produce high surface area for dye absorption. So once the surface area is high, so it will make sure the light is absorbed with the high amount, and so that the efficiency of the device can be improved. There are quite many materials that can be used as photoanode. The famous material is TiO2, SNO2, can replace TiO2, ZNO, niobium, Mb2O5, strontium titanate, SrTiO3, SNS2, anti-NIO, and we can prepare the photoanode with several morphologies. The morphologies refer to the shape of the particle of the photoanode. Okay. So from the left, top left corner, okay, the shape is nanosphere. Okay. In the middle, the shape is nanowire. Okay. The particle is the shape of nanowire. Okay. The third one, I'm not sure the third one. Okay. Honeycomb, yeah. The left corner at the bottom is a nanorock. Okay. The fifth one is nanotube. Okay. You can see the hollow part. Okay. It's called tube. And the last one is nanospider web. Okay. So the morphology of photoanode, okay, can be in nanospider web, and the efficiency depends on the shape of the particle of the photoanode. Okay, we move to sensitizer. Okay. So sensitizer have an energy gap separated by HOMO and LUMO. Okay. So the second component now is sensitizer or dye. So there are 4 cases I consider now. Okay. The first device has TiO2 only. The second one, RUL3 coated on TiO2. The third one, RUL2 NCS2 coated on TiO2. The last one, RUL dash NCS3 coated on TiO2. Okay. So I want to compare the performance of the SSC without dye and with dye coated on TiO2. Okay. It is found that the IPCE, incident photon to current efficiency, with TiO2 only, has the lowest IPCE, around 62%. Okay. But the IPCE with 3 dyes are higher than that of TiO2. Okay, this means that once a dye is spotted on TiO2, the dye improves the light absorption in visible region. Okay, this is the role of dye. Okay, TiO2 itself absorb light, but just absorb in ultraviolet region. Okay, the wavelength of ultraviolet is less than 300 nanometer, but the wavelength of visible light range from 300 to 700 nanometer. Yeah. So in order to fabricate efficient solar cell, the active material that is sensitizer must absorb as much as light in the visible region. Okay, it is pointless if researcher working on solar cell fabricate solar cell, but the cell absorb light in ultraviolet or infrared. Okay, it's not good solar cell. Efficient solar cell must absorb as much as light in the visible region, ranging from 300 to 700 nanometer. Okay, so in solar spectrum, the widest region is visible light. Okay. Okay. If we don't coat dye with TiO2, we get efficiency, but efficiency is very low. This is because TiO2 just absorb light in ultraviolet or ultra-low. Okay, so must understand the concept. Fundamentality must be strong before we fabricate solar cell. Okay. So again, the lower photograph shows the solar panel made of DSSC. Okay. The figure on the right shows the chemical structure of the three dyes mentioned. Okay. Next, I share several dyes, N3Z907 until C104. Okay, I'm not a chemist. Okay. I just use the material to fabricate solar cell. Okay, I'm here not to share with you about the chemical structure. Okay, I just want to share with you several dyes that we can employ in DSSC. Okay, so I use N719 in the lab at IMAN. Okay, and the best dye so far is N719, but its disadvantage is the price is very high. Okay, 3,000 one bottle. Kecil saja botolnya. Okay, 3,000. Okay, so the research is expensive. Okay, okay, we can have the dye material. Okay, JK46 until Indoline, but this is the short name. The IUPAC name of the material mentioned here are very long. Okay, we use short name of dye. Yeah, IUPAC name is very long. Yeah. Okay, next, I move to the third. Oh, sorry. It should be it should be the last. Okay, but mentioned at the third. Okay, is it okay? Yeah. So counter electrode of the SSC. As I mentioned earlier, platinum is the famous material for counter electrode for the device. Yeah. Since platinum is very expensive in price, so we just use platinum in little amount, okay, to dope into host material. Okay. And platinum can be prepared by various techniques: sputtering, electrodeposition, thermal decomposition, and etc. Okay. So platinum is quite expensive. Researchers in this field replace it with other materials such as carbonation material. Okay, example of carbonation material is carbon black, carbon nanotube, or graphene. And also conductive polymer such as PDOT, PSS, PDOT, PSS, PPY can be used to replace platinum. And also metal chalcogenide, cobalt sulfide also can be used to replace platinum. Okay, this is the third component, but I described the last. Okay. So electrolyte. Okay, there are three types of electrolyte: liquid electrolyte, solid electrolyte and gel electrolyte. So the efficiency of the SSC using liquid electrolyte is the highest, compared with the one using liquid and gel. But liquid electrolyte has the problem of leaking and stability. Okay, the electrolyte that I use in the lab is triiodide and iodide. And another liquid electrolyte is bromide, Br2, Br, SCN2, SCN negative, SECN2, SCN, and many more. So the example of solid electrolyte is solid polymer electrolyte. The ion mobility in solid electrolyte is lower than that in liquid electrolyte, so leading to lower efficiency. To overcome the problem, the researcher replace liquid and solid electrolyte with gel electrolyte. Gel electrolyte has no problem of leaking. And according to the literature review, the efficiency of the SSC using gel electrolyte is comparable with that of liquid electrolyte. So, whole conductor is another aspect of the SSC. It can be used to replace liquid electrolyte. So, if the researcher don't want to use platinum, the researcher can use other material such as gold, graphite, carbon-based material to replace platinum. If the counter electrode is no longer platinum, so the researcher can replace liquid electrolyte with whole conductor. The example of whole conductor is pyromyota. This example of whole conductor. So now I move to the SSC result that I obtained from my lab. So my PhD student focused on counter electrode made of aurum PD. This is a binary alloy. So the JV curve under elimination of 100 milliwatt per centimeter square light of the SC of aurum PD with various aurum precursor. The name of the precursor is HAuCl4 3HO 3H2O. So the variation parameter, the parameter that is override is the concentration of aurum precursor. So we start with the concentration of 0.10 millimolar, and the maximum concentration is 1.00 millimolar. The purpose of varying the precursor concentration is to find the optimal concentration for the aurum precursor. So it is found that the device using aurum PD prepared with 0.50 millimolar produce the highest GSC. And also it produced the highest efficiency. So the JV curve is analyzed to get GSC, VOC, and fill factor. So the photovoltaic parameter with various aurum precursor. So the device using 0.50 millimolar has the highest efficiency with the value 6.62%. So the device also has the highest GHC with the value of 17.07%. Okay. And the result is also compared with the device using a single metal, that is PD, and also compared with the device using platinum. Okay, we need to compare with platinum because platinum is standard counter electrode for DSSC. Okay. So the device using Aurum PD has the has higher efficiency, okay, than the device using platinum. Okay. But okay, and also it can beat the device using single metal, okay, PD. When we use PD only as counter electrode, okay, the efficiency is the lowest with the value of 4.14%. Okay. So the highest efficiency belong to Aurum PD, okay, base DSSC. Okay. And the optimum concentration is 0.50 millimolar. Okay. Okay, the efficiency of the SSC depends on 4 energy level because the SSC is made of 4 components. Okay. So it depends on LUMO and HOMO of photosynthesizer. In other words, it depends on the EG or energy gap of photosynthesizer, depend on Fermi level of TiO2, depend on redox potential of electrolyte containing the mediator of I3, I3 negative, and also depend on energy level of counter electrode. Okay, for this case, depend on the energy level of Aurum PD. Okay, finish the first performance study, that is JV curve. So from JV curve, we get 4 parameters: GSC, VOC, fill factor, and efficiency. Okay. And the efficiency must be verified by another parameter, okay, obtained from net characterization, that is electrochemical impedance spectroscopy. Okay, from this analysis, we can get RS, series resistance, RCT, charge transfer resistance, and also RCR, charge recombination resistance. Okay, theoretically, the lower RS and the lower RCT, the higher efficiency. Okay, but it's opposite to RCR. Okay. Theoretically, the higher RCR, the higher efficiency. Okay. Okay. Okay, the straight line in the Nike plot represents series resistance with the symbol RS. The first semicircle represents RCT. Okay. The second semicircle represents recombination resistance, and the last one, okay, is electrolyte resistance, represented by third semicircle. Okay. And also, researcher perform half cell measurement. Okay, half cell measurement is performed to get the exact value of charge transfer resistance because there are 2 interfaces in the device. The first interface is cathode electrolyte or counter electrolyte. The second interface is electrolyte photoanode dye, to get the exact value of charge transfer resistance. So half cell measurement should be tested on half cell. Okay. 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 Again, I show case study of a Nyquist plot of the device with various Aurum precursor concentration. Okay. The name of the precursor is SEUCl4 3HO2. Okay. So according to the Nyquist plot shown in figure 27, okay, there are 2 semicircles only observed. The first semicircle, as I mentioned just now, represents charge transfer resistance, and the second one represent RCR. Okay. So the Nike plot in the figure did not show the third semicircle. And the Nike plot is transformed into border plot to get carrier lifetime. Okay. So from EIS study, we can also get carrier lifetime. Okay. And the longer carrier lifetime, the higher efficiency. Okay. We look at the parameter, 4 parameter. Okay. RS, RCT, RCR, tau with various concentration. Okay. According to the JB curve, the device prepared with 0.5 millimolar yielded the highest efficiency. Now we need to validate the efficiency with the EIS parameter. Okay. Oh. Sorry, ada interruption. Itulah sangkut. Sudah lebih 1 jam kan? Saya, kenapa dia stop, dia masa sudah lebih kan? Okey, sekejap. Okey, ini delete. Sudah tadi. Okey, pergi pada the current slide. Connection is not stable. Kita tekan habis. Okey, ada lagi. Tadi ada lagi 20 slide lagi. Sekejap. Sekejap. Sudah berapa jam saya cakap? Sudah berapa lama? Tak apa, 1 jam lagi kan? Belum lagi kan? Belum lagi kan? Okay. Ini. Boleh tak? Masa balik ke? Share. Sorry ya. Okay, ini. Okay. Sorry, I continue. Okay, I continue. So the efficiency should be validated by EIS parameter. Okay. So the device with 0.5 millipolar has the lowest RS, 28.61 ohm, the lowest RCT, 4 ohm, okay. But the RCR is the highest, okay. And the carrier lifetime is the longest, which is the value 117 millisecond. Okay. Why cannot move? Cannot move. Connection tak stabil kan? Tunggu dia datang. Cannot move. Yeah.
To all participants, we have already sent the material, presentation slide through WhatsApp groups. You can check
Boleh,
through the file.
Ada beberapa minit lagi? Ada beberapa minit lagi? 20 minit lagi. Okey, ada lagi 20 slide lagi. Ya boleh ya? Okey. Okay, the next supporting parameter is IPCE. Okay. Okay. So the efficiency should be verified by IPCE. Okay. The higher IPCE is the higher efficiency. IPCE or EQE is defined as electron per second to photon per second. Okay, the current figure shows IPCE spectrum of the SDG using dye MK2. Okay. And the IPCE is flat. In the visible region, the highest IPCE is 80%, but its value is constant within the wavelength from 400 to 600 nanometer. Okay. Okay, case study. IPC spectrum with various layers of panchromatic dye. Okay, the name of dye is panchromatic dye, and the thickness of the dye is right. Okay, so the thickness is expressed in terms of the number of deposition from 1 time to 6 times. Okay, the result is quite interesting. The IPCE in the visible region increases with number of layers of dye. Okay. But in infrared region, the IPCE increases with number of layers of the dye. But we focus on the IPCE in the visible region, okay, from 300 to 700 nanometer. Okay. Okay, this is the result that my PhD student obtained. The IPCE with various aurum precursor concentration. Yeah. Okay, the highest IPCE is around 46%, belonging to the device prepared with 0.50 millimolar. Okay, so far the result is consistent. The IPCE is verified by RS, RCT, RCR, and also is verified by IPCE. Okay. Okay. And also IPCE can be obtained from the formula. Okay, 140 J photo multiplied by lambda, okay, I and C. And from this formula, from IPCE for each wavelength, we can get J photo. J photo is short circuit current. Okay, sorry, typo here, not density, just short circuit current. Okay, we get JSC for each wavelength in the whole spectrum. So we sum up J, we get JSC, short circuit current density. And the first way to get JSC is from the JV curve. Okay, that is the J-axis intersection. Okay, a lot of parameters need to related with efficiency. Okay, the second last is cyclic photometry parameter and TAFEL parameter. Okay, so I'm not going to explain in more detail because we are running out of time. Okay, case study again.
Usually we have 30 minutes more.
How many more?
Sorry, 30 minutes.
30 minutes.
Yes,
Okay,
yes.
plus giveaway, right? Plus giveaway, right? Okay. Okay. So what is the purpose of performing cyclic photometry? Okay, to get reduction current and oxidation current. Okay. So theoretically, the higher reduction current, the higher efficiency. Okay. Okay, we can get the J reduction at the lowest peak of cyclic photometry, and the peak, okay, at the positive J is called oxidation current density. Okay, so now we relate cyclic photometry parameter, okay, with efficiency. Okay, again, the device with 0.5 millimolar perform the highest efficiency. Okay, and it should be correlated with JPC. According to cyclic photometry parameters table, okay, JPC of 0.5 millimolar has the value 2.51 milliampere per centimeter square. Okay, this is the highest. Okay, and the delta E should be the lowest. Okay, delta E is the change in potential. Okay, so the lowest delta E, the highest JPC or reduction current are closely related to the highest efficiency. Okay, the last parameter is the parameter obtained from Tafel polarization curve. Okay. So we transform CV curve into Tafel polarization curve by using software. Okay. I did not do manually, we do using software. And the parameter from Tafel is J limitation and J naught. Okay. And there's a relationship between J naught and J limitation with efficiency. The higher J limitation and J naught, the higher efficiency. Okay. So the case study is consistent. Yeah. Sorry. The variation, the parameter variation is HAUCl4 3HO2. Okay. And we know that the device with 0.5 millimolar has the highest efficiency. Okay. And it is verified by the highest J limitation is 0.807 and the highest exchange current J naught with the value of 0.854 milliampere per centimeter square. Okay. Okay. Next, I continue with flexible di-sensitized solar cell. Okay. The flexible component is substrate. Okay. The flexible substrate here is PET coated on ITO. Okay. So flexible means we can reshape the device. We can reshape the device. And the performance parameter is the same. VOC, GSC, fill factor, and PCE. Okay. The device using PC25 perform the highest PCE with the value of 8.28%. Okay. It also has the highest VOC open circuit voltage with the value of 736 millivolt, the highest short circuit current density, 16.66 milliampere per centimeter square, and fill factor 67.5. Okay. So fill factor is very high, okay, above 60%. Okay. So the device using the counter electrode PC25 prepared on flexible substrate, okay, PET, perform the highest efficiency. Okay. Comparing with the device using platinum counter electrode, the device with PC25 has higher efficiency because the device with platinum has the efficiency of 7.42% only. Okay. And the device using single punning counter electrode just yield the efficiency with the value of 5.56% only. Okay. Then the parameter from EIS, that is RS, RCT, okay, related with efficiency. Okay, and just found that the device using P25 has the lowest RS, okay, with the value 10.2 ohm per centimeter square, and it also has the lowest RCT with the value of 19.8%. So the result is consistent. Consistent meaning that RS, the lower RS, the lower RCT produce the higher efficiency. Okay. Okay, this DSC retain high efficiency. Okay, so the efficiency dropped by 11.5% over 8 weeks. Okay. Okay. Next, I show the photograph of the counter electrode made or fabricated in my lab in IMAN. Okay. The dark region represent counter electrode made of palladium niobium, prepared on ITO. Okay. The transparent part is ITO. Okay. So palladium niobium was prepared on ITO substrate. Okay. And look at, very easy to fabricate the device using clamping method with clip. Okay. But clamping method is not actually good technique to fabricate device. Yeah. But this is what I have. What I have, clamping technique is suitable if the size of device is small. Okay. For your info, the size of device is around 1.5 by 2.5 cm, following the size of substrate. Okay. And this device produce the highest efficiency, if I'm not mistaken, around 6.7%. But the world record of efficiency of the SSC is 14%. Okay, it achieved around 44. Okay, 44%. Okay. Okay. And also the stability of the device need to be studied. Okay. So my student choose the duration of 30 days to study the efficiency, stability. Okay. On day one, the efficiency was 6.69%. Okay. After 30 days, the efficiency dropped to 3.87%. Okay. So the efficiency dropped by 40% or 60%. Okay. 60% drop. Okay. So this counter electrode made of aurum PD has potential to be developed as counter electrode material for dye center solar cell. Okay. Okay. Lastly, I would like to make summary on all type of counter electrode that I studied since year 2017. We look at the efficiency in year 2017 is very low, 0.09%. What do you understand by 0.09%? Less than 0.1%. Very low, right? But the material is not binary alloy. It is RGO. Okay, as year increase, the efficiency is also increased by changing the material. Okay, for our RGO, the PCE was 0.18%. Then the material was changed to argentum, RGO, rose to 1.3%. Okay. And the highest is 6.69% belonging to PDNB and PDAU. Okay. Okay. PDAU 6.69% obtained in the year 2023. PDNB got almost PCE obtained in the year 2024. Okay, PPY produced 3.08%. Okay, apart from binary alloy, my PhD student, my other PhD student also study other material than alloy and produce the efficiency which is lower than alloy-based DSSC. Okay, so 3.0% to 0.25. Once PPY was incorporated with graphene, so the PCE was improved a bit to 4.24%. Okay. So future work is to improve the efficiency by incorporating another material into binary alloy system to form ternary alloy. Okay, the example is PDRURS. Okay, the work on ternary alloy is still going on. Okay, thank you for your attention.
Thank you so much, Prof. Yusri. Please give applause to Prof. Yusri. Yeah. Thank you so much for the insightful presentation, Prof. Yusri. already give a material about the SSC. And in the beginning of presentation, Prof. Yusri also introduced about IMEN,
Yes.
University Kebangsaan Malaysia. Maybe some of you interest to study in Malaysia, you can also ask Prof. Yusri directly.
Yeah.
Okay, I will open the discussion session. Please raise your hand if you have any question. Mention your name, ask your question directly.
Okay, before that, I would like to mention that tomorrow I will present at physics department
at physics
about
department.
the SSC, research in
At
UKM.
Building
Okay.
B.
Okay.
Assalamualaikum, Prof.
Yeah.
My name is Shafinas. I'm from UiTM Shah Alam, Malaysia.
Yeah.
Okay, my background is biology, so I am very interested when you show me about the different colors of your dye. Okay, since I'm from biology, it triggers my mind and relate that with photosynthesis in green plants, where chlorophyll absorb visible light spectrum
Yes.
to conduct the photosynthesis.
Yeah.
That's why the green leaf appears green in our eyes. So my question is, before this you said that there's one dye that is quite expensive that increase the most efficiency in light absorption.
Yeah.
So I am interested to know what is actually the color.
What mean?
What is the color?
Color of the dye.
The color
Oh,
of the dye
okay.
increase
So
highly.
different dye has different color. Okay.
Yeah.
Different. Okay. There are two types of dye: synthetic dye and natural dyes. Okay. We can extract dye from part of tree, stem, leaf. Okay. But when we compare the efficiency of the SC using natural dye with that of synthetic dye, of course, the efficiency of natural-based dye is much smaller than that of natural dye. Okay, natural dye is ruthenium-based dye, such as N719, N7. Okay, the reason behind it, I myself don't know. Okay, but we have pro and con. If cheap, low performance. Do you agree with my view or not? If expensive, high performance. Okay. So I don't study at all natural dye in my research. Yeah. So we need to purchase. Okay. We have to purchase, but the price is very expensive. Okay, satu botol and seven mana itu sudah RM3,000. So kalau dapat restogram below 100K itu, it's difficult lah to do research. Okay. So we must get a big fund from government. Okay, government must support this field. Okay. So it should be above 100K. But we can try. We can try using natural dye. Daripada daun, daripada batang kan. Tapi we can expect the efficiency. It's very low. But for academic study, it's okay. You can publish in WAS journal, right? But to commercialize, I myself has yet to come to the level of pre-commercialization. Okay, I think, as I mentioned just now, it's not my academic role. It should be done by industry. Okay. Yeah. So you get the answer? You can try. People work on extracting dye from part of tree. Yeah. Daripada bahagian pokok. Okay. Tapi performanya rendah. Okay. The theory behind this is the absorption. If we compare the optical absorption of natural dye with synthetic dye, of course, the absorption in visible region, okay, produced by natural dye is much smaller than that of synthetic dye, such as ruthenium. Okay, did I answer your? Yeah.
So actually, what is the color? Is it green?
The color tu ada hijau, ada biru. Okay, saya pun tak pernah tengok lagi. Okay. Berpelbagai color, ada warna biru, warna hijau, ada yang panel tu, ini warna merah kan? Okay, ini saya tak tahu nama dye-nya apa. Okay. Tapi different color have different light absorption and produce different efficiency. Okay, lupa nama apa?
Shafinas.
Okay, as far as I know, the highest efficiency produced by natural dye is only 1% only. 1%. Okay, but the world record produced by the team in Switzerland is 14%. But they use synthetic dye. Okay. Okay. So my conclusion is don't study natural dye. Okay, even though it is cheap. cheap, low performance. Okay, we need to sacrifice. Okay, okay, use a lot of money to buy expensive material. Okay.
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 Thank you, Prof.
Okay.
The next question.
Okay, Assalamualaikum warahmatullahi wabarakatuh. I'm Ira. I'm from Sebelas Maret University. I'm from Department of Physics. I have a genuine question, Prof. Silicon solar cell have efficiency. Currently, we know that it ranges from 20 up to 25% for standard commercial. And from this presentation, it is said that it only maximum have 14%. So my question is, considering that, is there any special advantages from the SSC to justify continuing the development and research in this field? Thank you.
Okay. So I think I got the question a bit. Okay. You like to compare efficiency of silicon with the SSA. Okay. Is it your question? Is it your question? You compare efficiency of silicon solar cell with that of the SSA. Okay. We can still work on silicon solar cell. Okay. But in terms of academic, it's already died out. Okay, no more academic value from silicon solar cell. Okay. But for to make solar panel, okay, researchers use silicon solar cell. Okay, but it's expensive. Okay. Okay, coming back to the SSC, the highest efficiency is only 14%. Okay. 40% according to lab scale. Lab scale, you know lab scale? 1.5 cm by 2 cm only. But we cannot make prototype. The smallest size of prototype is 10 cm by 10 cm, right? So I have idea how to improve the efficiency of the SSC. We need to tandem. You know tandem? To combine with other type of solar cell. Okay, this is the only way. Yeah, but if you were to ask me, I will say it's quite difficult to commercialize the SSC because the efficiency is only 14%. Yeah, but when we compare the cost of the SSC with that of silicon, the cost is much lower. So pro and con, pro and con. Yeah. So that's why I just focus on academic matters, publish only. Yeah. So to commercialize, okay, we can try to make prototype. Yeah. But we must understand that when we enlarge the size of the device, such as the SSC, the efficiency drop. The efficiency drop. Yeah. So the efficiency in the lab scale is lower than the scalable device. Okay, compared with scalable device. Okay. But for prototype, we consider power, not efficiency, the output power. Okay, so this only that I can answer you. Is it? I answer your question already. Okay, you want to get my view right about this? So I will say has potential already, but when it become reality, I must say don't know. Okay, because the efficiency is 14%. Okay, among the third generation solar cell, efficiency has the lowest efficiency. Okay. The highest efficiency from third generation is perovskite solar cell, 27%. It's comparable with that of silicon. But perovskite solar cell has the problem of performance stability. Performance stability. Yeah. When we compare the stability of the SSC with perovskite solar cell, we find that the efficiency, the stability of the SSC is higher. So have pro and con here. Okay, the second pro and con here, higher performance but low stability. Yeah, the SSC has higher performance than, has lower performance than PSC, but higher stability but low performance. So pro and con. Okay, so we need to explore. Okay, we need to explore. Okay, just my view. Okay, the answer.
Thank you for the answer,
Yeah.
Prof. Yusri. Is there any question? Yeah. Committee?
Hello, Assalamualaikum. It's Khalid Hussain, doctoral student at UNS from biology department. So in my master research, my focuses was on the biodegradation and decolorization of textile dyes using bacterial consortia. So considering that fact, dye-sensitized solar cells utilize dye molecules for light harvesting. Is it possible to recover or transform textile dyes from wastewater into functional sensitizers for DSSCs? And what would be the main challenges regarding dye purity, stability, and photovoltaic efficiency in such an approach?
So you work on dye?
Yeah.
Natural dye or natural dye?
It was natural dye, yeah.
Natural dye.
Yeah.
So what's the application of your device? You don't study solar cell, right?
I just like remediate the textile dyes. That source comes from the natural.
So you degrade your dye?
Yeah, I
dye degradation, right?
Yeah, dye degradation.
Okay, what's the material that you use to degrade the dye?
I used anthraquinone based dyes.
Okay, so
Disperse dyes.
I think that you study photocatalysis, right? Or electrocatalysis? Because the application is different here. Okay, for my case, I use dye in solar cell, but in your work, if I'm not mistaken, okay, what I understood, what you mentioned to me, you degrade dye.
Yeah.
Dye degradation, right?
Yeah.
Okay, so the active material, I would like to know the active material that you use to degrade your dye, because your application is dye degradation.
Yeah.
So your dye natural dye.
Yeah, natural dye.
Natural dye. I think it's possible. Okay, it's possible. Yeah, but I don't study deeper because I have no experience with dye degradation study. But I think it's more easy to degrade natural dye than the synthetic dye.
Yeah.
Okay. Okay. I think possible. I think you can proceed with the work. Okay. From which university you from?
I did my master's in Karachi University,
In?
Pakistan. In Karachi University, Pakistan.
Oh, okay. Okay. It's possible.
Yeah.
Possible. Okay. Proceed with that. Okay. Okay. Okay.
Okay, thank you. We have still time, so any other question? Is there any question maybe about IMEN, maybe about your academic plan? We can use bahasa because we have similar language, yeah, Prof.
Okay.
You can use bahasa Indonesia, I think.
Boleh, boleh,
Prof
boleh.
Yusri will understand.
Okay, masa ada lagi? Masa saya dah habis belum? 1 jam setengah. Ada lagi?
Ya, masih ada 10 menit. 15.
Okay, tak apa. Kalau sudah tak ada soalan, so kita stop aja.
Okay.
Boleh tanya besok pun boleh. Besok saya jumpa lagi dengan mereka.
Okay. Okay. I think if you P6 student, tomorrow will be there will be general lectures in P6 department. Speakers Prof Yusri.
Ya besok.
Okay.
Okay, at 9 tomorrow.
Yeah.
Okay, waktu Malaysia, waktu tempatan lah, local time. Okay, di fizik.
Okay, I think we can close this session. Thank you so much, Prof Yusri, for the insightful presentation, and also all participants for your attention. We will have another session after this, so stay tuned. Thank you so much. Wassalamualaikum warahmatullahi wabarakatuh. Good afternoon.
Waalaikumsalam.
Thank you to our speaker and moderator. Please return to your seats. Ladies and gentlemen, we will now break for lunch and prayer. The program will resume promptly at 1 o'clock p.m. at the Academic Senate Meeting Room, first floor, Sutomo Darso Wiratmo Building. For those who wish to perform prayers, the mosque is located just across this building. Okay. You can enjoy the exhibition in this building, second floor, and then enjoy the lunch and will be served in this hall for all participants. Our distinguished speakers are kindly invited to move to the Academic Senate Meeting Room for preparation and a brief break before the program resumes. For all the participants, you can enjoy lunch here at this building. Thank you. We look forward to welcoming you back at 1 p.m.